Ever wondered why you share certain traits with your parents or siblings? The answer lies in a biological process known as meiosis. This isn't just any ordinary process, it's a fascinating journey of cell division that has a significant role in shaping who we are. Imagine it as a master weaver, intricately intertwining strands of genetic information to create a unique tapestry that represents you. It's this process of meiosis that ensures you carry a blend of your parents' traits, yet remain uniquely individual. But meiosis isn't all about inheritance. It's a key player in genetic diversity, a concept that's vital for the survival and evolution of species. By shuffling genetic traits, meiosis brings about an incredible variety of life forms, each equipped to thrive in their unique way. So, ready to dive into the captivating world of meiosis and understand its intricacies? Meiosis, unlike other cell division processes, consists of two parts, meiosis 1 and meiosis 2, each with distinct stages. Diving into the world of cell division, let's start with meiosis 1. It consists of four main stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. In prophase 1, chromosomes pair up and exchange genetic material, a process known as crossing over. Metaphase 1 sees these pairs align in the middle of the cell, while anaphase 1 pulls them apart to opposite ends. Telophase 1 wraps up with the cell splitting into two, and then comes meiosis 2. Like a sequel to a captivating movie, it too has four stages: prophase 2, metaphase 2, anaphase 2, and telophase 2. Here, the chromosomes don't pair up or cross over. Instead, they line up, get pulled apart, and the cell divides again, resulting in four unique daughter cells. Now let's delve deeper into each stage, starting with meiosis 1. During meiosis 1, the cell undergoes four stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Let's dive deeper into each stage, shall we? Our first stop is prophase 1. This is where the magic begins. The DNA in the cell condenses into visible chromosomes, each consisting of two sister chromatids. But here's the twist. In this phase, homologous chromosomes pair up and exchange genetic material in a process called crossing over. This crossover is a masterstroke of nature, ensuring that each offspring inherits a unique combination of traits. Next, we move to metaphase 1. The paired chromosomes, now called tetrads, align at the cell's equator. This alignment isn't random, oh no. The orientation of each pair is a matter of chance, leading to a mix-up of maternal and paternal chromosomes. This phenomenon, known as independent assortment, contributes to the genetic diversity of the offspring. On to anaphase 1. The tetrads split, and the homologous chromosomes, each with two chromatids, are pulled to opposite ends of the cell. Unlike mitosis, sister chromatids remain together. It's like a game of tug-of-war where the chromosomes are the rope. Finally, we arrive at telophase 1, the last stage of meiosis 1. Here, the cell divides into two, each with a random mix of chromosomes from both parents. The chromosomes uncoil, the nuclear membrane reappears, and the cell prepares for the next round. Meiosis 2. Interestingly, meiosis 1 is often called the reductional division. Why, you ask? Because it reduces the number of chromosomes from 46 to 23, preparing the cell for the formation of gametes, the sperm in males and eggs in females. And with that, we have successfully completed meiosis 1, but our journey doesn't end here. We've seen how meiosis 1 shuffles the genetic deck, creating unique combinations of traits. So buckle up for the next stop in our journey, meiosis 2. The adventure continues. Next on our adventure is meiosis 2. Mirroring the stages of meiosis 1, but with some crucial differences. Meiosis 2, similar to mitosis, comprises of four stages: prophase 2, metaphase 2, anaphase 2, and telophase 2. However, unlike mitosis, the end result of meiosis 2 is four genetically distinct daughter cells, each with half the number of chromosomes of the parent cell. Let's start with prophase 2. The nuclear envelope breaks down and the spindle apparatus forms. The chromosomes, each consisting of two sister chromatids, start moving towards the equator of the cell. But remember, these chromosomes are not identical. They've undergone crossing over in meiosis 1, swapping genetic material and adding to the genetic diversity of the offspring. Then we move on to metaphase 2. The chromosomes line up at the equator of the cell. But unlike metaphase 1, in which homologous chromosomes line up, in metaphase 2, it's the individual chromosomes that line up. This random alignment contributes to independent assortment, another source of genetic diversity. Next up, anaphase 2. The sister chromatids finally separate and move towards opposite poles of the cell. This separation is what distinguishes meiosis II from meiosis I, where homologous chromosomes separate instead. Lastly, we reach telophase II. The chromosomes reach the poles of the cell, the nuclear envelope reforms, and the cell undergoes cytokinesis, splitting into two. But remember, since meiosis II occurs in both cells produced in meiosis I, we end up with 4 daughter cells, each with a unique set of chromosomes. And there you have it, the journey through meiosis II. This process, while complex, is vital for sexual reproduction, ensuring genetic diversity and the continuation of life. Without meiosis II, we wouldn't have the genetic variation that makes each of us unique. Congratulations! You've just navigated through the complex process of meiosis II. With all stages of meiosis now covered, it's time to tie everything together and review what we've learned. Let's start with the big picture. Meiosis is a specialized type of cell division that results in 4 genetically unique daughter cells, each with half the number of chromosomes of the original cell. This process is crucial for sexual reproduction, as it produces the sex cells, or gametes, that are required for the formation of a new organism. We dove into the details of meiosis I, where we saw the cell go through 4 stages: prophase I, metaphase I, anaphase I, and telophase I. Remember, the key takeaway here was the exchange of genetic material between homologous chromosomes during prophase I, a process known as crossing over. This is what gives us that incredible genetic diversity we find in nature. Then we explored meiosis II, which mirrors the stages of mitosis. However, the outcome is quite different. 4 haploid cells that are genetically unique. That's one of the wonders of meiosis. It ensures every gamete is a one-of-a-kind masterpiece of genetic combination. Of course, we can't forget the role of meiosis in inheritance. Each of us is a blend of our parents' genetic material, thanks to the random assortment of chromosomes during anaphase I and the splitting of sister chromatids during anaphase II. This randomness is why siblings can look so different from each other, even though they share the same parents. In summary, meiosis is a beautifully complex dance of chromosomes, a process that not only creates diversity, but also continuity, allowing traits to be passed on from generation to generation. It's a fundamental part of life as we know it, and a fascinating subject to delve into. So, the next time someone asks why you have your mother's eyes or your father's smile, you'll know it's all thanks to meiosis. Now that we have a good understanding of meiosis, let's explore some exam tips and potential questions you may encounter. Firstly, to help remember the stages of meiosis, it's useful to create a mnemonic or a memory aid. For instance, you can use the phrase, "I play more tennis at the club," to remember the stages of meiosis: interphase, prophase, metaphase, telophase, anaphase, and cytokinesis. Visual aids like diagrams can also be beneficial. Draw each stage and label them. This not only helps you visualize the process, but also reinforces your understanding. Now let's discuss potential exam questions. A common question you may see is, explain the differences between meiosis I and II. For this, it's crucial to understand the key distinctions, such as the fact that meiosis I results in 2 haploid cells, while meiosis II yields 4 genetically unique haploid cells. Another possible question is, describe the importance of meiosis in genetic diversity. Here, you want to discuss the crossing over that occurs during prophase I, which leads to the exchange of genetic material and ultimately, genetic diversity. When answering these questions, always start with the basics. Provide a brief overview of the topic before diving into the specifics. This not only shows your understanding of the topic, but it also helps structure your answer. Also, practice, practice, practice. The more you practice writing out your answers, the more comfortable you'll be on exam day. Use flashcards to quiz yourself or explain the process to someone else. Teaching is a great way to ensure you truly understand the material. Lastly, stay confident. You've put in the work and you understand the material. Don't let exam nerves get the best of you. Take deep breaths, read each question carefully, and trust in your preparation. Remember, understanding meiosis not only helps you ace your exams, but also makes you appreciate the beauty of biology. Happy studying!